Hello student, I am Dr. Mamta, Assistant Professor in Hindu Girls College, Sonipat. I prepared this lecture under the aegis of Director General Higher Education Haryana for undergraduate botany student. The topic of the lecture is Cambium and its functions. The content which I am going to cover is listed as what is cambium, what is the origin of cambium, type of cambium, vascular cambium, its structure, its activity, factors affecting cambium activity, function of vascular cambium. A cambium is a lateral meristematic tissue that is responsible for secondary growth in plant. Secondary growth is defined as increase in the diameter of axis due to the activity of lateral meristem. A lateral meristem present on the lateral side of stem and root of a plant. A meristematic tissue are those tissues which undergo division in plant. These meristematic cells help in increasing the thickness of plant. Cambium is of two types, Cork cambium and vascular cambium. A vascular cambium is a lateral meristem which is differentiated along with primary vascular tissue, primary xylem and primary phloem from the procambium, responsible for secondary growth in plant through formation of secondary tissue. However, Cork cambium is also a lateral meristem, but it is also a secondary meristem. A secondary meristem means meristematic tissue originate from primary permanent tissue. Cork cambium is formed in the cortex region and it gives rise a protective layer known as periderm. So in this diagram you will see vascular cambium and cork cambium. Cork cambium is in the cortex region, whereas vascular cambium is made up of fascicular and interfascicular cambium. Vascular cambium. The vascular cambium have different characteristics. It is made up of single layer of living meristematic cell. It forms a cylinder of unspecialized meristematic cells in the woody plant. It does not transport water, mineral, food through the plant. Even it does not produce any lateral organ but form the woody tissue in plant. It gives rise to secondary xylem inside toward pith and secondary phloem towards outside, that is epidermis. The functional life of the cambium varies in different species and even in different parts of same plant. In a woody perennial plant, cambium is present throughout the life of plant. Vascular cambium is found in dicot and gymnosperm, but absent in monocot except few plants, for example palms and lily. Monocot did not show secondary growth, that is increase in the thickness due to the absence of vascular cambium. Vascular cambium is also known with other names such as main cambium, wood cambium and bifacial cambium. Vascular cambium is originated from procambium and procambium is developed from the derivative cells of apical meristem. The growing vegetative shoot apex have a cylinder of cells which is highly cytoplasmic, and these cells are known as residual meristem. A residual meristem have two portions, procambium and interfascicular parenchyma. A procambium is the more densely stained region of the residual meristem, and it is a primary meristem, give rise primary xylem, phloem and fascicular cambium. Some part of procambium get differentiated into primary xylem and primary phloem. Proto-xylem differentiate first in comparison to Proto-phloem originate first in comparison to proto-xylem. The undifferentiated region of procambium is known as fascicular cambium or simple cambium. The second part of the residual meristem is interfascicular parenchyma, and this interfascicular parenchyma give rise fascicular cambium through a process of dedifferentiation, means removing the defined function of a tissue. In monocot, complete procambium differentiate into vascular bundles, so there is no vascular cambium. In dicot stem, fascicular and interfascicular cambium combine to form a continuous ring known as vascular cambium or cambium ring. A fascicular cambium is present between primary xylem and primary phloem, and interfascicular cambium is present between two vascular bundles, lead to formation of vascular cambium. In dicot root, cambium originate partly from conjunctive tissue, a tissue present between primary xylem and primary phloem, and partly from pericycle. It lead to formation of complete cambium ring in root. There is difference between fascicular and interfascicular cambium. A fascicular cambium is originate in between or found in between primary phloem and primary xylem. And interfascicular cambium is present between vascular bundle. A fascicular cambium is primary meristem. However, interfascicular cambium is a secondary meristem differentiated from the primary permanent tissue. A fascicular cambium is derived from the procambium, and interfascicular cambium is derived from interfascicular parenchyma. Fascicular cambium is present in primary and secondary growth of the plant, and interfascicular cambium is present only in secondary growth of the plant. Structure of vascular cambium. A vascular cambium is made up of single layered of living cells which are highly vacuolated, have large nucleus rich in ribosomes. These cells shows plasmodesmatal connections, and they give beaded appearance due to the presence of pith. The single cambium layer is made up of two types of initials, fusiform initials and raised initials. Fusiform initials are tapering, elongated, give rise secondary xylem and phloem, whereas ray initials are small, isodiametric, give rise xylem and phloem rays. These initials divide periclinally and anticlinally. Periclinally means division parallel to the surface of the plant body. Now this is the surface of the plant body, and this division is parallel to the surface, and this type of division is known as periclinal division. If division occur perpendicular to the surface, this type of division is anticlinal division. Periclinal divisions lead to formation of stacks of cells, whereas anticlinal division increases the diameter of the vascular cambium. Fusiform initials. Vertically arranged cells in the vascular cambium is known as fusiform initial. Fusiform means spindle-shaped cells. Elongated spindle-shaped cells arranged in vertical row, and these fusiform cells produce secondary xylem inside and secondary phloem outside. Fusiforms and its derivatives form axial system in plant, and these cells lie parallel to the surface of the stem. These cells seem fusiform when seen in tangential section. If we cut the section of stem in this way, this will give us a tangential section. The product of the fusiform cells is xylem, secondary xylem and secondary phloem. The secondary xylem is made up of different type of vessels, tracheids, sclerenchymatous fiber and parenchyma. And phloem is made up of sieve element, sieve cells, sclerenchymatous fiber and parenchymatous cells. Fusiforms cells are arranged in two forms stratified or stored arrangement and non-stratified and non-stored arrangement. When the cells are of similar size and they lie parallel to each other and end at the same level, this arrangement is known as stratified or stored arrangement. This arrangement is considered as more advanced phylogenetically. It developed gradually from the non-stored arrangement. In non-stored arrangement, cells are of different sizes and they did not end at the same level. The stored or stratified arrangement give rise stored wood after division, the example of which is tamarix. The non-stratified arrangement is more common, longer than the stored type, and they give rise non-stored wood. Example is rust. Rays initial. Rays initial are arranged horizontally in the vascular cambium. They are much smaller in size. Isodiametrically, their size did not show much variations and they produce phloem rays and xylem rays. These initials mostly involved in storage and other sideway transport. These ray initials present in uniseriate, biseriate and multiseriate depending on one, two or more longitudinal rays of cells. If these rays initials are present in xylem, they are known as xylem rays, and if they are present in phloem, and they are known as phloem rays. If we look on the vascular tissue in three-dimensional way, the elongated cells represent the fusiform cells, isodiametric cells represent the ray initials, fusiform cells give rise secondary xylem towards pith and secondary phloem towards outside. Activity of cambium. The activity of cambium is divided into two stages. Formation of axial and radial system by periclinal division and dilation, increase in circumference of cambium by anticlinal division. Axial system is consists of secondary xylem and phloem and it is formed by fusiform cells. Fusiform cells divide periclinally, lead to formation of two daughter cells and one of them get differentiated into secondary xylem. The other cell again divided lead to formation of again two cells and one of them then differentiated into secondary phloem. The meristematic fusiform cells repeat this process again and again, lead to formation of secondary xylem toward pith and secondary phloem toward epidermis. Initially, all cells look similar and this will give a multi-layer appearance to the cambium, and this zone which consists of cambium, xylem and phloem is known as cambium zone, but cambium is always single layered. Formation of radial system. A radial system is represented by medullary rays and these rays are present in xylem and phloem. They are of multiple thickness and height. Secondary medullary rays are derivative of rays initials and they divide periclinally, forms parenchymatous rays. These radial systems involved in the radial conduction of solute. The second process in the activity of vascular cambium is dilation. Due to the expansion of the secondary xylem, cambium also increases in size to maintain its continuity, and this is done by anticlinal division or radial division. The process of increase in diameter of cambium cylinder is known as dilation. Factor affecting the cambium activity. There are various factors which increases the cambium activity. For example, plant hormones. Indole acetic acid and gibberellic acid activate the cambium activity and lead to formation of more wood in plant. Day length also affect the cambium activity. In short day plants, there is formation of narrow and less tracheids, whereas in long day plants, there is formation of broad and more tracheids, and this is due to the more accumulation and synthesis of auxin in long day plants in comparison to short day plants. Gravity also affects the cambium activity. In the branches of gymnosperm and angiosperm, a special type of wood is formed due to the gravity, and this wood is known as reaction wood. In gymnosperm, this wood is formed on the lower side and it is known as compression wood, like compression wood here, whereas in angiosperm it is formed on the upper side and known as tension wood. The unequal portion of the wood is due to the difference in the synthesis and accumulation of auxin, and we know that auxin synthesis and accumulation is affected by gravity. The compressed wood shows more lignin deposition in comparison to the normal wood. Function of cambium. Cambium is responsible for secondary growth in plant, that is increase in the thickness. The primary tissue is made up of primary phloem, primary xylem, cortex, epidermis and pith, whereas in secondary tissue there is increase in the thickness and it is made up of secondary xylem, secondary phloem and cork cambium. Determination of age of plants. Due to the cambium activity, there is formation of growth ring in plants, and these growth rings are correspond to the age of the plant because they are formed each year. By counting these rings, we can analyze or we can calculate the age of the plant. Role in horticulture practices. In angiosperm, budding and grafting is possible due to the presence of cambium. In grafting, a portion of stem that is known as scion is grafted on the root stalk, whereas in budding, a part of the bud is grafted on the root stalk. When these scion and rootstock join, they will lead to formation of undifferentiated mass of cell known as callus, and this callus then differentiated into xylem and phloem lead to formation of vascular tissue on both xylem and root, on both scion and rootstock. This form a continuity of vascular bundle, and once the continuity exists between xylem or maintained between scion and rootstock, it will behave as a complete plant. But these budding and grafting is not possible in monocot plant due to the absence of cambium. Healing of wound. Cambium is responsible for healing of injury or wound in plant through the formation of wound tissue. When injury occurs in the plant, the parenchymatous cells form an undifferentiated mass of cell known as callus or wound tissue. The outer layer of the wound tissue develop thickening and form periderm, separating the lower part healthy part of the plant from the infection through compartmentalization, means isolating of the older injured tissue from the healthy tissue. Cambium in monocot. In few monocots, cambium is present, for example Dracaena and Yucca. In these plants, there is formation of vascular tissue in the cortex region, whereas in root, this cambium layer is formed in endodermis. Palm did not show any type of secondary growth, but it showed increase in thickness due to the gradual increase in size of cell and intercellular spaces, and this type of growth is known as long continuing of primary growth. So from this lecture we have learned that what is cambium, what is the function of cambium, its structures and different factors which affect the cambium activity. These are few questions from which you can revise this topic. Define interfascicular cambium, define axial and radial system in plant. What do you mean by cambium? Explain its origin, structure, function. Difference between fascicular and interfascicular cambium. Difference between fascicular, fusiform and initial rays. Write a short note on activity of cambium. Explain how cambium help in wound healing. This is all about cambium and its function. Thank you.